Hello, everyone. In this short video, we're going to discuss transport mechanisms and the transport of small molecules, in particular in the cell. And the reason we care about it is because a lot of energy goes into these processes. We saw previously how what's the energy currency in the form of ATP. Well, as I will make the point again and again in this video, about a quarter at least of eukaryotic cells' energy reserves are used to transport small molecules in and out of it. And that makes sense thermodynamically because cells are open thermodynamic systems, as we have discussed. They have a cell membrane, and that is the cell's boundary. And of course, the cell exchanges both matter and energy with the environment. Okay, and so this is where this exchange takes place, and this is why the transport mechanisms are very important. So not only the energy, but also the number of different chemical reactions that as a fraction of the total that a cell uses to do this business, to carry out this business is huge. Again, maybe as high as a quarter or a third of all chemical reactions. So within some membranes, of course, for a eukaryotic cell, you have the outside membrane, the plasma membrane that separates it from the environment, but also inside the cell, you have many membrane-bound organelles. Transport through those membranes can also be highly regulated. The exceptions seem to be, although even they are dysregulated, but for small molecules, the nucleus is not really a significant barrier because the nuclear membrane is not a significant barrier because there are large nuclear pores that even small proteins can go through. Likewise, the outer mitochondrial membrane does not really present any obstacles for small molecules because, again, the pores are are very large, very wide. Now, the inner mitochondrial membrane is a completely different story. Not even a proton can go through. So that's separate. In addition, even for the tightest membranes, some small molecules can go through. And the list is shown here on this slide. You have water, oxygen, carbon dioxide, ammonia, and ethanol are pretty much, they are movable across all biological membranes. So the last one, pay attention to it because whenever people drink and they are intoxicated, essentially ethanol will go everywhere in your body. So how can we move small metabolites across a membrane barrier? Well, usually so there is a membrane barrier, barrier shown here in this dotted lines, and you have some carrier protein, okay, shown here in gray. The metabolite will bind the carrier protein, so it cannot go through the membrane bilayer, so it needs some help with proteins. And it will bind, let's say, on the outside, or on this left side of the membrane barrier, and then something in the conformation of the carrier will change, and all of a sudden now the metabolites shown here in red will be presented to the other side of the membrane bilayer. Okay, so that's typically how things move. So let me repeat again. There is going to be a carrier protein of some sort that will help the metabolite to move in and out. We'll see there are many different ways that this can happen, and depending on how much energy you use or not. But there is always going to be some conformational change of this carrier protein, okay? That will, in the end, will result in the net transport of the metabolite from one side to the other. All right, so if no energy is needed, this transport mechanism is passive. So typically what happens in this case, the metabolite moves down its concentration gradient, so from a high concentration to low concentration. So that does not require energy. However, if energy is needed, then we are talking about an active transport mechanism. And typically, again, the metabolite moves against the concentration gradient. So of course, it's not thermodynamically favorable, and you have to provide the required energy. So we're going to discuss these different categories of transport mechanism. We have facilitated diffusion, antiport, symport, and transport with ATP hydrolysis. So passive, as the name implies, again, as I said, no energy is expended, and we've seen already how no carriers are involved for some small molecules, such as oxygen, carbon dioxide, and so on and so forth. But for all the others, it could be passive, so you may not need to spend any energy, but still you need the carrier protein. So with exception of this small, like gases usually, oxygen and CO2 or water, the others still need a protein. However, you may not need to spend energy because the metabolite moves down its concentration gradient. It's very rare, actually. This is a quite rare mechanism, except there is a very important example and exception, so if you wish. So glucose actually comes into our cells through facilitated diffusion. It's a passive mechanism. So this is an extremely important example of passive facilitated diffusion. Our cells do not need to spend any energy to bring glucose in. So how does that happen? Well, because it's passive and it moves across, it moves down its concentration gradient. Of course, on the outside, when you have a big meal, there is a lot of glucose, and then you bring it in. So it doesn't cost the cell any energy currency to bring the glucose in. How does that happen? Well, you can envision this carrier protein shown here in gray that just spontaneously flips in these two different conformations. And let's say this carrier protein has a cleft that something can bind to it, like such as glucose. Okay, so when it just happens that this binding site, this cleft faces outside, okay, then glucose will bind in. It will essentially just hitch a ride and wait until the conformation flips. So now the binding site is on the inside. The glucose molecule will come out. There is no concentration. There is not enough glucose in the inside of the cell, so it will come off, and that's how it comes in. Now, of course, if you keep bringing an enormous amount of glucose inside, you may build a higher concentration of glucose in the inside. So then glucose will come out actually and equilibrate, right? If that's a possibility. So that's a classic example of facilitated diffusion. The mechanism, as I said, is rare for most metabolites. It doesn't usually happen, but it does happen for glucose. And given the role of glucose in biology and metabolism, that's an extremely important example. Why does it not happen? Because organisms typically compete for limited resources. Okay, so non-concentrative uptake is not a good strategy, right? So it's just too hard to envision such a mechanism for many other metabolites. All right, so it turns out that transport mechanisms can be described with the same equations that we have seen before, okay, for any other reaction. So you define the concentration, you have a reactant on the outside and your reactant on the inside, and the ratio of the concentrations is the equilibrium constant when you have the concentration inside on the numerator and outside on the denominator. So that's when the two concentrations are equal, okay? Then you've reached equilibrium. So that's the equilibrium constant, 1. Okay, super simple. And you can use the same fundamental equations. You can have the delta G's and the delta G zeros, all the standard free energy changes associated with the transport, and you can write it just like any other chemical reaction. And we keep going, we simplify this equation as we've seen before. And in the end, all you really need to find the free energy chains, okay, associated with the transport is to just measure the concentration in and out and plug it into the equation, and you got your free energy chains associated with the transport of this metabolite A. Just play with it if you want. It's kind of interesting. Our cell, it's actually very important, our cells that line up the inside of our stomachs, right? So we all know that the stomach juices, the gastric juice is extremely acidic. That's the whole point of, that's the purpose of the stomach, and really acidic. So the pH is 1, okay? But the cells that line up the inside of the stomach, of course, there are normal cells, so they have the inside, their inside has a pH of almost neutral, pH 7, so almost 0 protons, okay? So free protons. However, they have to pump all these protons outside. So what's the free energy difference required for this transport of protons, right? It's an astounding large number if you calculate it, and I'll give you the answer. So these cells, I will not give you the exact answer. All I'm going to say is that these cells have this pump that pushes pH, that pushes protons out of the cell into the stomach, and they have to spend an enormous amount of ATP to do this, okay? Because again, you take protons from where the concentration is low and you put it where it's high, and you keep doing this, so they go against their concentration gradients. Protons want to come in from the stomach into the cells that line up the stomach, not the other way around. So imagine the amount of energy that these cells have to spend to do this, to acidify the inside of our stomachs. So the idea here is that also that this you can envision this again in the same way, this transport phenomena, to that you have to expend some energy getting this. Imagine that this is the equivalent. Disregard this useful work part for a minute. We'll get back to it, but this is pretend that this is the stomach cell, let's say, that lines up the interior of our stomachs. So it has to spend a lot of energy to get these protons, to get them out where the stomach is, right, the inside of the stomach. Now, in a different situation, which is really what happens in our cells, in our mitochondria, when we make ATP, building this concentration gradient, in effect, this is what this is. So lots and lots and lots of protons, let's say, outside, and those want to come back in, right? So if you find a way, as our mitochondria are doing, actually, and we'll discuss this in the second part of the course, to couple this gradient building, okay? So they build this gradient, and then because these protons want to come spontaneously, want to move in, you can couple it with useful work, okay? And that's actually the basis of making ATP in our mitochondria. As we will see, there is a transport phenomenon there, so you're resulting in a net. You don't actually spend any energy in the case of mitochondria. You'll see how that happens, but there is a net transport of protons. And then when these protons have to come back in, that's when the ATP generating machine is stuck in the membrane. So it couples the movement of protons back in with useful work. Okay, so transport phenomena are very important for that too. All right, now we'll talk about antiport, a variety of other mechanisms. As I said, glucose is unusual, but in most cases, what happens, you want to move something in where you want it, you better couple it with something else that moves in the opposite direction. For example, it doesn't have to be the opposite direction, but in this case, it is the opposite direction. So you couple a favorable movement with an unfavorable one. Favorable meaning moving down the concentration gradient. So you have one moving in one way, the other one the other way. One is against its gradient, the other one is down its gradient, and then the two can be coupled. How does that happen? So we don't have facilitated diffusion like we had with glucose. Instead, you have two binding sites, one binding on the one side from the outside, the other one from the inside, and you go through these transitions, okay, the conformational changes, and in the end you present the two binding sites in a different configuration, out and in, versus when you started with, and that's how you couple the two. Schematically, this can be drawn like this. And again, I know I'll go over a little fast with equations, but you can see the slides at your own pace. The idea is, like, again, you can describe these reactions, these transport reactions, as any other chemical reaction and do exactly the same equations as we have seen before, and come up with the numbers. And in one case, so again, this summarizes really what we got here. We have in an antiport, one of the metabolites moves in a passive way. So it moves down its gradient. So you don't need any energy to move that one, but you couple something favorable with something unfavorable, like the B metabolite here, and that's how you succeed in the end to move ATP, from the outside, let's say, to the inside. Symport is exactly the same principle, except they both go in the same direction. Okay. But it's usually, again, the same fundamental principle. It just changes the relative orientation of where one versus the other moves. But it's exactly the same principle. Okay, one down its gradient, and you couple that with another metabolite that moves against its gradient. And just go again fast, but it's the same idea. All right. Now, the last one is kind of is extremely common, okay? And this is where a lot of ATP is. So in the previous examples, the antiport and symport, we did not spend ATP, okay? We just used the built-in, let's say, energy for the metabolite that moves down its gradient and coupled that with a metabolite that needs to move against its gradient. What if you don't have this option? So what if you just want to move a load from one side of the membrane to the other side of the membrane, and you don't care, you're just going to spend whatever. You're going to go to the bank, your ATP bank, and spend ATP and do it. So here what happens really is that you couple ATP hydrolysis, which triggers a conformational change on the carrier protein. So they both bind on the same side, let's say ATP inside, let's say in this case, ATP and the other guy, the green metabolite, and the ATP hydrolysis provides the energy necessary to do this conformational change. So now the binding site for your metabolite finds itself on the outside, and that's how you moved it. That can be extremely costly. I mean, you need at least one ATP, to bind and be hydrolyzed. So you consume a lot. For every molecule, let's say, you want to pass across, you have to consume at least one ATP. It's very costly, and this is where a lot of your cell's energy go. And for example, for very simple things like sodium and potassium, they are transported with such systems in animals. Their concentration is kept so tightly, it's so highly regulated. I mean, every time you go for a blood work, they will measure your potassium levels, let's say, and they're so highly regulated within it's extremely tight. And why? Because then if they have to, otherwise you have major heart problems, for example, how your heart will beat and so on and so forth. So the cells, our cells are extremely, all eukaryotes really, very careful in regulating their intracellular concentrations of sodium and potassium, and they are prepared to pay for it, let's say. So even when you are resting, a third of ATP consumption in a resting animal is consumed in this process. So it's huge. We're not going to talk any more about it, but since we're talking about metabolism, this is it's astounding. I don't know, when I saw these numbers, it's actually quite astounding. We keep talking about ATP and all these other things, and then if you ask, the logistics, where does it go? And to find out that the third goes into this simple reaction, I mean, it's incredible. So we will close at this point. So we covered all the introductory topics, and in the next series of videos, we're going to start with glycolysis. Thank you.